the recording, and I'm going to first start with introducing Dr. Pepato, because I'm not sure if all of you are familiar with his works. And then Dr. Pepato will be free to start his presentation. All right, so Dr. Pepato graduated with a Bachelor in Biological Sciences from the Institute of Bioscience of the University of Sao Paulo in 2002. He then obtained a master's degree in 2005 and a doctoral degree in 2009 in zoology from the same university. Since 2011, he is a professor at the Universidade Federal de Minas Gerais in the Department of Zoology. I apologize for the pronunciation. Dr. Pepato is also a curator at the Archaeological Collection House by the Universidad Federal de Minas Gerais, sorry. And between 2017 and 2018, he was a postdoctoral visitor at the University of Michigan here in the US, where he collaborated with Dr. Pavel Klimov. His interest is on marine mites, and he used them as model to phylogeography, led him to a broader interest on cariform phylogeny, and his research aims to infer the evolutionary past of marine mites by using molecular biology to target and infer biographical patterns of coastal regions around the globe. So according to Dr. Pepato, alacaridae are the ideal model for better understanding of regional biography due to the lack of dispersive stages. All right, so with that, it's all yours.
Well, thank you very much. Good afternoon to everybody. I start apologizing because I'm not that skilled with languages. So I apologize in advance for my poor English. I will talk a little bit about some news on interesting, at least to me, a very interesting clade of mites that is compounded by Halacaroidea and Parastegona. Just a moment. We had some delay. So I will pass through these topics. I will provide some context about Halacaroidea and Parastegona. I will talk a little bit about previous ideas on thrombiformes, classification, and phylogeny, with special attention to Halacaroidea and Parastegona. I will talk about our article on among realms, land, freshwater, and sea transitions, and the intriguing consequences of recovered topology of Halacaroidea Parastegona. And I will talk about ongoing research, including analysis with additional terminals that change a little bit the results from this study, and talk about some methodological concerns that were raised during our work. So we are not the ones who choose a group of animals to study, but usually the group is who choose us. So I started studying marine mites because by the late 90s, a research program on characterization, conservation, and sustainable use of biodiversity, inspired by the Rio 92 Convention, started in São Paulo State, and researchers started massive and intensive surveys of marine terrestrial fauna and flora in this state. And this marine biology laboratory, the SEBIMAR, housed the samples from marine organisms, along with other marine institutes in the state. And as undergraduate student, I volunteered to help in sorting the material. And marine mites started accumulating without anyone who were able to study them. And it motivated me to start studying them. So it was something by chance and happened, and so we are here today. The marine mites, they occur from the upper littoral to abyssal depths. These grayscale pictures, they are from littoral species from the intertidal zone. These others, the Batellacarus, pictures provided by Dr. Furka, came from thermal vents from depths larger than 4,000 meters below the sea level. So we have marine mites occupying all depths in the oceans. They are very easy to distinguish from other prostigmata because they have a reduced idiosomal taxi. The cheliceraal digits, fixed cheliceraal digits reduced, similar to Anistina and Parasitegona, something shared with many other, and Elenterogona, something shared with many other prostigmatas. The pulp is linear without an evident thumb and claw complex, and the pulp usually is four segmented. Superficially, they are similar to some snoot mites, especially some Cunaxidae, and maybe it is why they were traditionally grouped with Bideloidea, with snoot mites. But as we see, they have nothing to do with them. And I became interested in this particular family mainly because they seemed to me, as Emily said, a great model for biogeography and phylogeography because they don't have different from many marine species. They don't have a dispersive planktonic stage that may be transported by the sea currents. They usually are sluggish, small animals that live that like to be very attached to the substrate, like algae, colonial animals, or among grains of sand. They are usually classified in a superfamily named Alacaroidea, with a small, much less diversified freshwater family named Mite-y, restricted to Australia. And this family was included in Alacaroidea mainly due to the overall similarity of their gnatosoma with the gnatosoma of some Halacaris. Parasitegona, on the other hand, they are much more diversified. We had much more species described in this group. Most of them, we have a single family that is an exception. Most of them have a larvae that are parasitic and heteromorph relative to delta nymphs and adults. So they have a complex life cycle that includes usually a parasite, but even more usually heteromorph larva, calyptostatic protonymph, and then a free-living predaceous delta nymph, a calyptostatic tritonymph, and then free-living adults, predaceous very similar to the delta nymphs. A problem that we face all the time here is that we can't associate immediately larvae and adults, and the taxonomy of Parasitegona is based on both. So we risk having, and sometimes we have, species, the same species described as larvae and as adults, and sometimes it takes long years to match them and synonymize. We're approaching indeed this in our lab by sequencing barcoding markers to associate adults and delta nymphs with larvae, or hearing the larvae to become delta nymphs, or females to lay eggs that hatches in larvae, to describe the entire life cycle, or at least all active instars. It is something that we are doing here. About the previous ideas on Trombitiformes classification and phylogeny, with special attention to Halacaroide and Parasitegona. Traditionally, Acariformes are divided in Trombitiformes and Sarcoptiformes. Trombitiformes include a small group with two families, Spherolichida, and extremely diversified group named Prostigmata. Here, Spherolichida usually is regarded as a basal lineage, and Prostigmata is traditionally divided in Anistina, Eupodina, and Eleuterogona, despite that in some references you may find some kind of variation around these names, around the determination of these names. Usually Anistina includes Parasitegona and some families such as Anistidae, Edycacaridae, Adamistidae, Tenerifidae, and Seculidae. Eupodina traditionally included Alacaroidea, also snout mites like Bideloide, Bidelidae, and Conaxidae. Eupodoidea including Antideoidea, Labdostomatidae, and Eryphioidea, that are very important worm-like mites that feed on plants or upon vegetables. And Eleuterogona includes Calicoidea, Rapinatoidea, Tetranychoidea, Heterostigmata, and may or may not, depending on the author, Paratideide, Pseudocaridae, Pomerantizoidea, Stimocaridae, that are less diversified families. There is in older literature some incongruence among who is the sister group to Parasitegona and Anistidae. Some consider that Eleuterogona is the sister group of Parasitegona and the families included traditionally in Anistia or Anistina, because they share a thumbical complex well developed and the reduction of the fixed chelicerial digit. Concerning Parasitegona, we have a handful of hypotheses, sometimes considering the terrestrial Parasitegona as a monophyllum, usually with calyptostomatoida that are large mites when adult, with gnatosoma that may be retracted into the idiosoma, along erythroid that includes Ismaridida, that has a very similar condition regarding the gnatosoma that also may be retracted, or at least in the other erythroid, in erythraide, the chelicera may be retracted. So this group comprising from stoma and erythroid share this ability of retracting at least the chelicera. While the other terrestrial Parasitegona usually are grouped together. But these are just examples. We have a lot of debate about these placements. Here we have other example that is especially interesting to me because it places, for example, Chiseroide as basal among the group that includes trombiculoid and trobigioid. And in this case, the terrestrial Parasitegona is not regarded as a monophyllum because Hydracarina is recovered in the middle between erythroid and calyostomatoidea and the trombidina. Well, from 2015 onwards, molecular data started to tell us something about these relationships, and in this relatively small sampling that we could perform in 2015, interestingly, we recovered labidostoma basal. That was something that many archaeologists already thought by this time. Indeed, in the 2000.
Good catch. Sorry about that. I think it's my fault. Sorry.
It's okay. So labidostomatidae was recovered as a basal lineage among trobintiformes instead of the spherolichid that was sampled, and Alacaridae was recovered as sister group to Parasitegona. So the traditional Eupodina exploded, blown up. And it was also recovered in the next year by this much more comprehensive regarding sampling of Parasitegona article by Dabert and colleagues, Dabert, Rita Proctor and Dabert, where also halacaroide, in fact, was recovered as sister group of Parasitegona. And Labdostomatides or Labdostomatidae was recovered as a basal lineage of Thrombidiformes. The relationships among the main lineages of Parastegona verdis across models and partitions employed. But it's interesting that they recovered a small lineage traditionally included in Thrombigene, Stigotrombidoidea, as sister group to freshwater Parastegona, and Stigotrombidoidea indeed is freshwater, so it would imply in a single transition from terrestrial to freshwater in Parastegona. So last year we completed a considerable sampling effort targeting into the transitions among realms, among land, freshwater, and sea, using 5 genes, the ribosomal 18 and 28S, the CL1, DHITSP17, SRP54, that these two later are protein coding nuclear genes, and CL1, it's probably is well known by you. It's a mitochondrial protein coding gene. Here the co-authors, Dr. Klimov, Mark Harvey from Australia, and our PhD candidate here, Samuel, who studies mainly Parastegona. And we sampled 150 families, 367 terminals. We obtained a large sample of groups like Brachypelina because they are very good as calibration points for time tree inference, since we have a good fossil record of Brachypelina. Eupodina, Anistina. We include as many families of aquatic mites as possible. Additional terminals for informative paleontological calibration points, and outgroups without taxa previously at some moment of history mentioned as possible sister groups to Acariformes, like Pseudoscorpion, Solifuge, Palpigrid, Palestiformes, and Riccinulae. Here we have a list of freshwater terminals. You see Stigotrombidae, Pesidae, and many Hydracnidia and freshwater Halacaridae. And marine including Pontarachnid, that is Hydracnidia that moved to sea. Many Halacardies, some Oribatida, and Astigmata that also are Oribatida. Labdostomatidae came basal again instead of spherolithic. And it recovered a large clade comprised families traditionally included in Anistina and Eleuterogona, something that is congruent with ideas of Lindequist in 1996, where he points to this relationship between families traditionally included in Eleuterogona and those traditionally included in Anistina, plus Halacaroide, in fact. Indeed, Teneriffidae, that's a family that looks totally Anistoidea, It was recovered along Eleuterogona as a basal offshot of Eleuterogona, and shares an intron that is presented by most of Eleuterogona. That's something very interesting, like a molecular apomorphy. The freshwater Stigotrombide was recovered basal across all analyses. The basal Parastegona. But most of arrangements could not be rejected by our tests. And examining the transitions among helms, we noticed, and it's why we called the paper one-way ticket to the blue. It is a song from the 50s, but also it's what we observed because we have most of transitions from land to sea or freshwater. And sometimes we have transitions from land to freshwater and then to the sea. And we almost don't have transitions or potential transitions from aquatic to dry land, like from sea or freshwater to the land. And we found it interesting. We don't have cases of we have almost no cases of this kind of transition from aquatic to land. The single exception may be in Parastegona, and it is very interesting because the basal lineage we recovered in Parastegona is from freshwater, as in the sister group that is Alacaroida, because the basal lineage in Alacaroida also is from freshwater, and it makes likely that the common ancestor was from freshwater. That is something very intriguing to us. It is not intuitive. And we also noticed a very old transition from the sea to freshwater in halakite mites that comprises some genera that may be very abundant in some freshwater environments, like Poralacarus, Porolohumanella, Limnolacarus. Well, what we are working right now, what we are doing now, we are eager to move to new generation sequencing, but we still have some things to do with Sanger. And we recently could expand our sampling with more terrestrial Parastegona. We included some families that were missing from our data set, like Chisaridae, Neotrombidae, Echimenotrombidae, Trombellidae, Neotrombidae, e especially special acquisition that was Allotanalpodidae. That is a very interesting family from New Zealand and the southern portion of South America. Also Podotrombidae, Erythreidae, Smaligidae, Calyptostomatid that were present in the former analysis, but we could improve the sampling. And some additional Hydracnidia and a special family, special genus in Halacardi that is the freshwater genus Lobalacarus. Why Allotanopodidae is so special? It's because it differs from other Parastegona in having larvae and delta nymphs and adults similar. They are not heteromorph and they are all free living. So they don't have the same life cycle as other Parastegona, as most of Parastegona. We have some reversions in some groups, but to free-living larvae, but it is clear from the larvae morphology itself that it is homoplasy, it's a reversion. In this case, it is clear that the animal lost not just the parasitic condition of larvae, but also that the larvae is morphologically similar to the adults and delta males. This new analysis messed with our previous hypothesis. It is advice to you all don't keep pet hypotheses because they die. You lost them. It's terrible. It is sad. Because Stigotrombidae was not recovered as since a group of all other Parastegona, and Allotanalpodidae was recovered as sister to them, but with mild support, and most of relationships we recovered in Parastegona, indeed, they were not so well supported. They were not recovered by our new analysis. But something interesting appeared in analyzing terrestrial Parastegona, because we have, as expected by Velborn, for example, in 1991, some families were recovered as basal among Trombidina with considerable support. So we have a group of Johnstonianidae, Neotrombididae, Trombellidae, and Chizeridae. That is sister to Trombiculoidea and Trombidioidea, and it is somewhat expected and very interesting. And curiously, Lobolacarus didn't group with other freshwater halacard mites, so we have two ancient incursions of marine mites into the freshwater, continental waters. Concerning the common ancestor of Alacaroida and Parastegona, apparently we have still a high probability of a freshwater ancestor, a freshwater common ancestor of Alacaroida and Parastegona. And some things must be investigated because we know that some of those basal Trombidini, they need a lot of moisture to complete their development. So it's something that must be considered, because when coding, the helm in which the mite lives, we are maybe too strict. We couldn't take into account that some depends upon a lot of water to complete their development. So I have some methodological notes, some things that our work led us to think about, and I would like to share with you. we have this old tree by Darwin, right? And he shows that he was able by this time of tree thinking. But in this case, the tree is just the relationship among species. We have the interest, or we have this dream of having a much more complete picture of how evolution occurred. In the origin of species, he shows something that is a research program that is much more complete, because he introduces a time scale and the diversification process into the tree. And I think that everybody is much more interested in this, in knowing how the history occurred, not just the relationships among species, but also when and how the things occurred, and is in it that I go into now, because we still are sampling our terminal and sometimes our sequences, having as criterion of selection the some kind of systematic problem. Oh, I want to know if some group is monophyletic, or if this guy goes with this other. And we don't use to choose our sampling design solving this big picture, this history of life. We are accumulating fast genetic information about mites. Indeed, there are some ongoing projects doing new generation sequencing, but we are not going to solve this big picture just by accumulating sequences. And it is good because it is a task in which any archaeologist having even a small budget or just a microscope can contribute to. Because the current methods that for phylogenetic inference, they blur the phylogenetic tree inference with other aspects, like the time scale of evolution and the diversification process. For example, in our article, we use a program named Beast to infer the time tree. And this program, Beast, it employs a model of sequence evolution, a model information about the age of divergence through fossil calibration that may assume different shapes. In our article, you use an unbalanced sample of our knowledge about the fossil record, because you see we have much more calibration points from the Cretaceous, and it is not true regarding the fossil record of mites. We don't take advantage from all information of the new fossils, because we don't have sampling enough to put them in a tree. And we don't have morphological studies enough to precisely place the fossils to use them as calibration points. So in this age of genomic scale sequencing, one of the most important tasks is doing the good and old morphological phylogenies, because morphological phylogenies is what allows us placing correctly the fossils, to use them to infer time trees. And even with sampling just of morphological data, we can do inferences on the timing of diversification. Like in this article by Klimov, that describes some fossils of Paratid age. And something very important to finish my talk is some of the time tree estimations, they push the root of the tree and the ages to very ancient ages. And it just looks to be a bias of the models that are employed. The time tree inference in programs like this, they use models of diversification that are or you pure birth, death, models of diversification. And apparently in real world studies, diversified groups, they push the root to ages that are related to the central tendency of the models, and not with the dynamic that occurred. Here we compare two lineage per time plottings that show this bias in our own analysis. You see that applying the birth model, we have age that is much older for the root than in other approach to time tree, and it may be related to the bias introduced by these models. So the conclusion from the experience of working with this group, with this all this molecular data, with this attempt of solving the dynamics of transitions among helms, is that face the problem of current methods is related to a program of research that you include thinking about them, pay attention to what is really running in phylogenetic inference programs, and maybe pay some attention in solving individual families and trying to guarantee for these families evenly distributed sampling that may help in making these programs behave correctly. And finally, I think that even moving to new generation sequencing, we do not need disregard the accumulated data of morphology, mitochondrial genomes, and markers obtained by Sanger, because any progress in science has a cumulative aspect, and we must be able to correctly evaluate the previous who came before us to make real progress. So it is it. And thank you, and thank you for who along this last few years supported our research.
Thank you, Dr. Pepato. Does anyone have question in the audience? You can either chime in or just write it in the chat. Feel free, question or comments. Nobody? All right, I can ask one. I'm more you finished by saying that we still have to mix the morphology, I mean take into account morphological character with sequencing, and I was wondering how exactly how does this mite breathe if they breathe in the water? I know it's totally a different topic of what you presented, but I'm interested if they move back from the land to the sea and then the sea to the water, how they morphologically adapt from that. Did they had a different breathing system? Do you know about that or not?
The morphological adaptations related to these transitions.
Yeah.
Well, I think that one of the most important things to look at is the genital acetabula and epimeral pores, because they are related to the to osmoregulation. These organs, they are pumps of ions into the hemolymph of mites, or in some cases they are used to kind of drink water, like they are used also to absorb water. And for example, Hydracnidia usually have multiplied genital acetabula or epimeral pores. The same is true for halakard mites that moved from the sea to fresh water. And even halakard mites that live under influence of fresh water, but live in the sea, like in near to rivers, they also have the genital scapula external, and so I think that it's the most important. There are some others that are largely under studied. For example, when I was studying the sperm cells of marine mites, we noticed that the osmolarity of marine mites are much higher than usual among mites. So, we needed to add a lot of salt or sugar to set the osmolarity. And it shows that there are some physiological adaptations that probably you are ignoring because they are understood. But there are some others, like most of aquatic, they lost the trachea, but well, some Hydracnidians don't, but in the case of halacardi, they don't have the stigma, they don't have the tracheal system.
Okay, thank you. Thank you for your answer. In the chat, we have a comment from Heather saying that just a comment, not a question. Great talk, Almir, and very nice to know that morphology still has an important role to play in studies of mite phylogeny.
Thank you. It's so nice see you here. Thank you for your kind words. It's yeah, and it has a very important role still. And I if you are if you can't afford right now sequencing because it is very expensive, you still can contribute to the large picture of mite evolution because there is a lot to do with morphology and our own hope of placing correctly the fossils among living extant mites is through morphology. So we must keep here the 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 classical studies on morphology and try to improve our knowledge on morphology by adding our own observations. And for example, something that makes me kind of upset is that there's few uh, uh, researchers interested in doing internal morphology of mites. We had a great colleague who we miss, that was Gerd Alberti, that was very productive in observing, describing the internal morphology of mites, and yeah, we need more Gerds. We need more information about the internal morphology. He alone provided so many sites into mites, and it's the kind of, and we now have we have a much better technology. We can do a lot of observation that was impossible a few years ago. And it's something to think about.
All right, does anyone has another question? All right. We had a comment from Ed Mohammed Khan. Thank you so much, dear Dr. Pepato, for such a nice and comprehensive talk. My question is there any morphological problem in identification, especially in species or marine mites? What kind of morphological problem a taxonomic is facing?
Well, I consider marine mites relatively easy to work with. The taxonomy is not that complicated. They have reduced ketotaxy. So if compared with most of Prostigmata, they have usually 5 pairs of dorsal sitting, and it is we can decide about species identity mainly do their external ornamentation, this kind of feature. But now that we are accumulating more and more sequences, we are detecting some cryptic species. So we have the same problems as any other animal group, that we have species that can't be correctly identified by morphology at all, because the differences are are not those we are used to. They are not those we are used to include in species descriptions. For example, in a study we are finishing here in our lab, we noticed some pairs of cryptic species along Brazilian coast in marine mites. And probably the main differences are physiological, are ecological, because they are related to different environmental conditions. So I think that it's a relatively easy to work group, that you face the same problems as any other group of animals.
All right, thank you for your answer. I think we're about to finish here since we are reaching our time. I thank everyone who came today, and I hope you enjoy Dr. Pepato presentations. Just a quick reminder, we will have two more a professor speaking for this webinar, this 2023 webinar. We will send over email throughout the years. There is one scheduled in October and one in November, I believe. So please stay in touch and keep track on your email. And thank you for being here, and have a nice day.